Let's go through the cell quantification and quality control parts of the NPX workflow. As mentioned, now we need to count the cell concentrations in order to accurately aliquot the chosen number of cells for exonucleus, PCR, and sequencing. It's important to note that the amount of cells that you're aliquoting will directly impact the read requirements for sequencing. During the greater part of the protocol, it's fine to use automated cell counters, such as K2, Luna, or the Countess. It's only at the end of the protocol, when cell concentrations will be low, that automated cell counters are not reliable and you need to perform manual cell counting. We do not recommend using a dilution factor, since that will dilute your samples even more, which can increase the risk of counting errors. Remember that counting fixed cells can be quite tricky and time-consuming. So take your time and don't rush this step. When you count using a hemocytometer, we recommend that you use these 4 corner squares in order to achieve a good average. Perform the measurements in duplicates and use the average to calculate the cell concentration. So this is how you calculate the cell concentration if you don't use any dilution factor. So now that you have the cell concentrations, you're ready to aliquot your samples for exonucleus treatment and PCR amplification. After PCR, your products will be stable at -20 degrees Celsius. Next, it's time to do quality control to make sure that you have the expected product size. But before you do quality control, you need to perform 2 rounds of ampere beads cleanup so that you get rid of any unused PCR primers. Quality control can be carried out using gel electrophoresis or any type of fragment analyzer, such as the tape station or the bioanalyzer. Once the QC results confirm that you have the correct product, qubits should be carried out on your samples so that you can accurately measure the DNA concentration. The only thing that's left to do now is to pool and prepare your samples for sequencing. And if you don't have your own sequencing instrument, you can always use your closest sequencing facility. Once sequenced, our Pixelator software will help you with data analysis and tutorials to really dig into the data and answer your specific scientific question. For the first time ever, you will now be able to add multiplex spatial information like polarization and colocalization to the cell surface proteome of single cells. We really look forward to all of the novel insights that this will bring to biology and stages of health and disease. You're always welcome to reach out to support if you have questions of any sort. Thank you for watching.